Hello and thank you for tuning in to the next in the JCI series, Conversations with Giants in Medicine. I'm Ushma Neal. I'm lucky to have the opportunity to speak with James Allison of the MD Anderson Cancer Center while he's here in New York to collect the Lasker DeBakey Clinical Medical Research Award in honor of his discovery and subsequent development of monoclonal antibody therapy to unleash the immune system to treat cancer. His work on the T-cell antigen receptor, CD28, and CTLA4 have formed the foundation of immunological research and also are the next great hope in cancer treatment. Thank you so much for joining me today. I'm delighted to have a chance to speak with you. Can you start by telling me a little bit about where you grew up?
Yeah, I grew up in Alice, Texas, a very small town in the Rio Grande Valley. Everything outside when you walk around had needles or would bite you or stick you or something, but pretty harsh environment. But it was a fun place, and I got interested in biology and things like that. I was lucky enough to have some good teachers who, I must admit, it was a pretty bleak place to grow up, but I had some good teachers who pointed me towards better things, science and medicine.
What about your parents? What did they do?
Well, my dad was a country physician. He did house calls and was paid in tamales a lot. My mother died when I was very young of lymphoma, and so I grew up mostly with my dad and in some foster homes and things. But
did he ever impress upon you that he would love to see you follow his footsteps into medicine?
Yes, he did. He would take me on house calls a lot, and he never really forced me to think about going to medicine. But it was there. I understood that he wanted me to. But I was lucky enough to have some research experiences due to some teachers of mine that got me involved in summer programs at the University of Texas at Austin when I was in high school, and did lab work, and I just loved it.
Well, what kind of lab work?
Oh, at the time it was just figuring out how much iron yeast needed to grow optimally, But it was hypothesis, and you tested, and it was great. And I started college, with a pre-med major, but I started thinking about it, having watched my dad deal with patients, and I realized that doctors have to, physicians have to really be correct all the time. They have to, diagnose what's going on, pull out an algorithm that they've memorized and apply it to the patients, and they've got to be right. And in science, what I learned was you don't have to be right. You have a hypothesis, and you test it, and you only have to be right some of the time. And I was not sufficiently disciplined or rigorous or whatever to think about being a physician. I thought that being wrong a lot was a lot more fun, and so I decided to become a scientist and go that route, because you only have to be right, as I said, occasionally, and you should be wrong most of the time, because if you're not, then you're probably not on the edge where you ought to be, testing.
Well, you say that, but you graduated from high school early and started college at age 16. So clearly you were a good student that was right a lot of the time. So did you have that intellectual curiosity from the beginning? Did you ground your interest in science from those summer programs? Did you ever think about English or languages or anything else?
I never really thought about. I wrote poetry for a while and really liked reading a lot. But no, I wanted to be a scientist. From the time I was a junior in high school or so, I was toying with it, and by the time I was a freshman in college, I said, I've got to be a scientist. I want to be a scientist. And
so what did you end up studying while you were at college?
Biochemistry, which was a good way to start, because it teaches you precision and quantitation, and as I learned when I got fascinated by the immune system, I learned that there wasn't much precision, because immunology at the time was a lot of thinking. I mean, people had just discovered that there were T cells, and there was a lot of debate. I mean, the professor, Bill Mandy, that taught me immunology in the undergraduate course wasn't even sure that there was such a thing as T cells, much less as whether they had a specific receptor that could recognize things. And even when I went to Berkeley in the early 80s for a faculty position, we had worked out the structure of something I said was the T cell receptor, but people, argued whether that was correct or not. But I think that my training in biochemistry really equipped me well to.
So when in this trajectory did you abandon medical school or abandon being a pre-med major and decide to do a PhD?
I think it was my freshman year when I was taking organic chemistry, and I don't want to say this incorrectly, but organic chemistry can be beautiful if you get into it. But the way it was taught, the particular course that I had, was just memorize and regurgitate, memorize and regurgitate. And I think they intended the course to really be the cutting course. That if you couldn't take it, then you shouldn't be a doctor, and it was taught that way. And so that was one thing I really regret, that I missed what could have been, the beauty of organic chemistry.
It's the only class I ever got a C in as well, so I feel you.
Yeah, well, I did worse.
So at this point you decide to do a PhD. Then when did you fixate on what you were going to study?
Not really until my first faculty job. I wanted to do immunology as exposure to it in this undergraduate course that I had. But also, at the time I was working on asparaginase. It's a complicated story. I felt that I had to do medically related research in order to avoid going to Vietnam, because I was, at the age and whatever, and being drafted. So I started doing more medically oriented research.
And so did it work?
Yeah, asparaginase. Well, it was an
Well, I meant the dodging of the draft, but tell me about asparaginase.
The draft or asparaginase? The asparaginase worked, actually. Dodging the draft did, but it was a little more difficult. But anyway, but asparaginase was being used at the time to treat children with leukemia, because a lot of childhood leukemias lack the ability to make asparagine, so they require it, and it's made in excess quantities in people, and it's in the plasma. And so if you deplete it with an enzyme, I mean, I didn't show this, other people did, but we were trying to come up with a better enzyme to inject into people to drop it. And so, that's what my PhD thesis was about. But one of the things that I did just because it seemed interesting, after I cured mice of lymphoma with this enzyme treatment, I decided to re-inject them with the same tumor, and they were immune. And I was reading about this stuff from Lloyd Old and others that were talking about tumor transplantation agents, but I was just startled. I said, these mice now, I treated them with this drug that just deprives them of an essential nutrient, and yet they're immune. And so that was in the back of my head for a long time. I didn't really work on cancer directly for a long time, but that really impressed me. And that, I couldn't do that now, because I'd have to propose the experiment to the IUCUC and get it approved and all that. But this was just a one-off. I was in the lab, and I'll just inject these mice again. And
so let's go back a little bit. You are working on these mice with asparaginase. You've managed to stay in the States as opposed to going off to war. Is it at this point that you transitioned to do a postdoc?
Yes, I really wanted to go from being a biochemist into becoming an immunologist. So my professor that I'd taken the immunology course from helped me get into a laboratory at Scripps Clinic, which at the time was one of the best places for immunology in the world. And so I went to the laboratory of Ralph Reisfeld there. But my project was purifying MHC molecules for amino acid sequencing to try to figure out the structure. it really wasn't immunology, it was just biochemistry again. And so it was a little frustrating because I got to see immunology going on all around me, but I was doing big biochemistry.
But with another student, you kind of did some side experiments.
Yeah, with another postdoc, we did some side experiments and how tumors are recognized by the immune system. And my professor actually said, we showed him the paper, and he said, well, I don't want my name on it. You guys just did it. So anyway, he submitted it to Nature, and it was published. So just by 2 postdocs without the PI, it was pretty interesting, and we got a lot of notoriety for that.
And then at this point, you transitioned back to Texas. There's something about Texas that brought you back. What was it?
Well, I'd lived in Austin. I grew up in Alice, but I spent my formative years in Austin, and there was a lot about Austin that was really fun. I mean, one thing that I think most people understand is the music scene there, and Willie Nelson had just moved there, and J.J. Faulkner, and Stevie Ray Vaughan, and it was just a lifestyle that I really enjoyed. So I heard that they were opening a research lab that MD Anderson was operating in near Smithville, Texas, 7 miles from Smithville, Texas, which was a town of only 3,000 people. But it was about a 30 to 40 minute drive from Austin. So I heard they were opening this, and I went back and I said, hey, you guys need me. And so they hired me, and that was good.
So what was that like, starting your first independent lab? Did you have an idea of what you wanted to study and what kind of resources you were given? Was it a struggle? Were you able to get students and fellows?
It was actually my experience there was quite good. Largely, I think, because at the main campus they forgot about us, and so I was free to do whatever I wanted. And so I was working with a good team of people. The initial project was to make monoclonal antibodies to. There's rats that are fed carcinogens. The livers undergo stereotypical changes, and so we wanted to work out the molecular mechanisms of that in the surface molecules that change. And so my project was to make monoclonal antibodies, and that was fine. But I still want to do immunology, and I didn't consider that. I considered that using immunology to answer a question. But then, at the time, the burning question in immunology was, what do T cells use to recognize antigen? And I, without going into all the details, I drove from Smithville to Houston, the main campus, to hear Irv Weissman give a talk, and Irv was very prominent, still is, immunologist in those days, and he gave a seminar, and I had one of these aha moments during the seminar, and I had an idea of how to identify the T cell antigen receptor, and went back to Smithville and sort of dropped everything else and launched into this, and we succeeded in my lab. And I had a lot of support, though. From MD Anderson, I had a lot of support, but they basically let me do whatever I wanted. Anyway, I just worked out the structure of the T cell antigen receptor, and then went to Irv's lab.
So you took a sabbatical for a year in his lab.
Tried to clone the genes, and we cloned a lot of stuff, but it wasn't,
And around the same time, Mark Davis was working on this as well.
Yeah, Mark did it independently. The only thing that Mark, got from our work is that there were 2 chains, and both of them had constant and variable regions. So, he did it by a completely independent method. But still, it was the first time, I went from small time, lab. There were only 6 faculty at the Science Park at Smithville at the time. And
so here you are, you come from this small place, you've done this major work in identifying the T cell receptor. You've got Irv Weissman peeping in your ear, especially if you've done the sabbatical, that you need a bigger stage. Yeah. So at this point, you are recruited to Berkeley, yes?
Yeah, so while I was in Irv's lab, Lenny Koshland, the late the late Bunny Koshland, asked me to come to Berkeley and give a seminar, and I did. And about a couple of weeks later, they offered me a job, and I was an assistant professor at MD Anderson. They offered me a full professorship at Berkeley, and I thought, But still, I was a little bit scared. You know, it was intimidating. You know, the big science, the whole Stanford thing. You know, and I was happy. You know, we had a really collegial. You know, everybody helped everybody else work. You know, at Smithville, and I took a long time to make a decision. And finally, Irv called me again and said, "What do you want to do, Jim? In 20 years, you're going to sit on your porch, say I could have been a contender." You know, and I said, "Yeah, you're right. I got to go for it." You know, so I accepted a job at Berkeley.
So you transitioned to Berkeley. You've got the TCR to your name, but it's a completely different environment. So was there culture shock in moving
tremendous
there?
culture shock. I mean, you know, we at Smithville, I mean, it was good. I mean, people cared, and there was a lot of teamwork, a lot of esprit de corps. You know, let's all work together. But when I got to Berkeley, it was like, wow, the intellectual environment was just incredible. You know, the students, the faculty, everything was just, you know, up here somewhere compared. Yeah, so it was a little scary at first, but.
So you faced a faculty that thought that the most basic problems were the ones that were the most important to answer, as opposed to an entire system of the immune system.
Exactly. There was, at the time, Dan Koshland was reorganizing the biology at Berkeley, but there was a lot of sentiment there that you should be working on first principles. You know, how does DNA replicate? How is RNA, you know, made into protein? You know, things like this, very fundamental things. And if you were working on mice as opposed to worms or flies, you know, you were considered suspect already. So there was a lot of, much less, you know, I was interested in the immune system and interested in cancer as well. And yeah, it was a bit of a fight there, because my science wasn't pure. It wasn't first principles.
But it's during this time you then started to develop your ideas about CD28 and activation and deactivation signals of the immune system.
We'd worked out the structure of the antigen receptor, and other people, you know, really showed that it was right. I took a purely structural biochemical approach, and there were other people that had taken more of a functional approach and confirmed that the molecule that we had done. But then there was some really incredible work done by Ron Swartz and Mark Jenkins and others at the National Institutes of Health in the late 80s that said that you know, it wasn't self, non-self, you know, where you just flip a switch and the immune system goes. You know, they showed there's co-stimulatory signals that were required, additional signals. And so I became interested in what those were. And so, you know, a lot of people have done work with the molecule called CD28, and that sounded like the right actor to be the co-stimulatory signal. But the work that had been done was more like saying, oh, well, if you hit this molecule, you get more proliferation, IL-2, whatever. But the key thing about co-stimulation was there's a second signal that you have to engage in addition to the antigen receptor to get a naive T cell to go. And that was what really I wanted to answer. But so we, others, Brian Seed had cloned the gene for CD28, human gene, so we got some sequences from him and cloned the mouse version and expressed it in cells, made monoclonal antibodies to it, and showed that engagement of that was sufficient and necessary as a second signal.
At this point, were you thinking about any applications of this, or were you purely interested in the fundamental immunology?
At that point, it was just purely fundamental immunology. No, I wasn't thinking of any applications.
Okay. So start the story about CTLA4 then.
Well, when we cloned CD28, and we weren't the only ones to do this, a lot of other people did. We put the sequence in the gene bank and said, what's related? And there was this molecule called CTLA4 that Pierre Goldstein in France had identified a few years earlier. And all he knew about it was that it wasn't, I mean, it's misnamed, it's in all T cells after activation, but that was the key. It's not in resting T cells. It's only expressed after activation. And because of the sequence, particularly the ectodomain, the extracellular domain with CTLA4, seemed just cried out, this has to do something like CD28. And so a lot of people, again, we weren't unique in that, went after it. What does this do? And so the first person to really get insight into it was a guy named Peter Lindsley, who was in Seattle. And he, the first thing he did was to make a recombinant form of the molecule and show that it bound. By then, he had already, Peter had shown that CD28 binds to these molecules called B7 that are pretty much restricted to dendritic cells and other really good antigen-presenting cells. And so then he made a recombinant form of CTLA4 based on the sequence to show that it also bound to those same molecules, but bound them with an avidity, 2 logs higher than CD28. And so he made an antibody to it and threw it into cultures with T cells being activated with antibodies to the T cell receptor to CD28, and threw in antibodies to CTLA4 and showed that it increased proliferation. So he concluded there was another activating costimulatory molecule. And so we did the same experiments, but I won't go into the details, but the thing that you had to add it in soluble form rather than cross-linking form. We got the same results. We did the same experiments. He got the same results, but the fact that you had it in soluble form and it increased worried me, because most antibodies block, they don't activate.
So you had the heretical idea that instead of being an activator, it was an inhibitor.
Exactly, and Jeff Bluestone, just to be fair, at the same time had the same idea independently. And
so you two are the lone wolves, crooning that perhaps this is a break.
Yeah.
How long did it take for people to actually accept that idea?
I think it was probably close to 3 years until Tack Mack and Arlene Sharp and the late Cynthia Chambers from my lab made knockout mice, and they basically turn into a big lymph node because their T cells can't stop dividing. And so it was realized that it is an essential downregulator, because until then, people thought that, it's obvious that when you get a T cell response, you go from a few dozen, few hundred T cells to 10 million in 3 or 4 days, and to eliminate whatever it is, you got to stop that. And so the idea was, at the time, was activation-induced cell death. All the T cells just die. So and that's not incorrect. That does happen, but we had the idea there's this limiting thing that stops co-stimulation after a time, and so T cells just sort of grind to a halt.
So at this point, I think I remember reading you had some students who had grown tumors in mice and injected them with CTLA4 antibody.
Yeah.
And you couldn't believe the results. You asked them to do it again, but it was Christmas break.
Yeah. So the postdoc Dana Leach did the experiment, showed me the results, and I went, whoa, this is. I expected it to slow tumors, but the mice that got the CTLA4, he showed me these graphs, and they were cured, basically all of them. And I went, oh, this is important. And there's a couple of reasons. One is he wanted to go see his girlfriend, but another one is I thought, well, we have to do this blind. And so I said, we'll set up the experiment again and I'll measure the tumors. And so he went off to Europe and I measured the tumors, and it was kind of depressing because they all grew. And so for the first several measurements, there was no difference between the groups. But then at some point, I think it was like 2 days after Christmas, when I was measuring them, one group had sort of flattened out, wasn't growing anymore. And then as I followed, again, I didn't know what was what. But then these went away, just went away, and the others, we had to euthanize the mice. And at that point, I said, oh, this is
big.
This is big, because we did nothing to the tumor.
So it's still in mice at this point.
Yeah.
And Bristol Myers Squibb held a patent saying that they thought CTLA-4 was an immune stimulant. So they were not about to make a humanized antibody to put into people.
No.
So what was the process like of then actually getting a pharmaceutical company, which you ended up with Medarex, to develop a humanized antibody?
Well, I spent almost 2 years going around and talking to big pharma and some small biotech firms and trying to instrument the ideas and there's a lot of skepticism. I mean, the data were the data. By that time we had done it against many different types of tumors and many strains of mice and shown that it was generalizable. It would work either by itself or we could add, a vaccine or something if it didn't work by itself. And there's a lot of skepticism, and the fact that Mr. Myers Squibb already had a patent also put people off, because I'd say, but their biology's backwards. They got it backwards. And the comment I got was, well, they've got a lot of lawyers, and you don't want to, you're. I think the word that was used was dirty. Your IP is dirty. It's not clean. And I thought it was clean. So I was at a Gordon conference and was just presenting, again, 2 years on, all the data, saying, I don't know why we can't move this forward. And by then I'd gone to Rick Klausner, who was the head of the NCI at the time, and said, Rick, I can get grants and I can study this and I can, but we need to be able to treat cancer in people, so the NCI ought to get involved. And, you're interested in just funding clever grants or just in curing cancer? And so they actually funded the RAID program, Rapid Access to Interventional Developments. But as it turned out, I became ineligible for that because this company, Alan and Pippa Merrick, convinced Larry Gold, the head of Nexstar, to actually take this project on to make aptamers. And the aptamers didn't really, they couldn't make this RNA molecule, but they tried very hard. But after a while, we made an antibody. And then, anyway, what happened was Alan also knew a guy named Niels Lundberg, who had a company, it was originally called GenPharm, and then was bought by Medarex, became part of Medarex, that had mice that had human immunoglobulin genes instead of mouse. So they made fully human antibodies. And so they said, okay, we'll take this. And so they made an antibody, MDX-010, which became ipilimumab.
So at this time, you've got ipilimumab. Harold Varmus comes calling and says, come to MSK, Memorial Sloan Kettering, and you would have the benefit of actually watching ipilimumab go into patients.
It was a little more complicated than that because I realized I knew nothing about the clinic. Nothing. I was so naive. But I knew that the biology of this was different than a cancer drug, which kills cancer cells, and it's evaluated by killing cancer. You treat patients, and if they grow in the face of the drug, then it's a failure. And yet, in our mouse experiments, the tumors regularly grew in the face of treatment, but then they'd be rejected. So by then I was totally committed to seeing this go into humans. And so I started looking around. I looked at several places actually. But Sloan Kettering, for the reasons you said, kind of resonated. So I chose to go there because, New York was where, in the area, Medarex was there, Mr. Myers Squibb, all the drug companies. So it's kind of, I decided to go there and learn about the clinic and, and so make sure that nobody hurt my baby, basically.
And you watched your baby grow and take its first steps all the way to FDA approval. I mean, this must have been tremendously gratifying to see and watch as the first trials within melanoma were positive, that it wasn't necessarily regression of tumors, but it was overall survival.
Exactly. Yeah, and the trial started off being designed to be progression-free survival, but they began to see patients that tumors would grow and they would go away. And so, BMS and Medarex had the knowledge by then to change the endpoint of the trial, which some other companies didn't, that had other anti-CTLA-4 antibodies. Yeah, so that was really cool. And but still, in a way, at the time it was still abstract. I mean, I'd had a lot of cancer in my family, and so I knew what that was, but that wasn't really why I was doing the work. I was doing the work because I wanted to understand T cells, but when I realized what we could do with this information, then I, became really committed to making sure nobody screwed it up. And so I had that opportunity at Sloan Kettering, and I had this very emotional moment, and I think it was 2006, when I got a call from Jed Walchuk, a clinician there, who said, come down to the outpatient clinic. And so I said, why? I'm busy, because the outpatient clinic's at 53rd and 3rd, I think, and I was at 68th and 1st, so I said, okay, well, I'll come down there. And anyway, I walked into the room where he was, and he was there with this woman, Sharon Belvin, who was 22 at the time and had been diagnosed with metastatic melanoma and failed everything, and basically was hospice bound, and had a round of treatment with anti-CTLA-4. And I met her when she'd come back a year after she'd been declared tumor-free. And I walked in the room, and she was with her husband and her parents, and she was celebrating. She said, I want to meet the guy that invented this. And it was a big hug fest, and everybody started crying, and that's when I thought, this is important. And then about a year and a half later or so, I got an email from her with a photograph of her first baby, and then a couple of years later of her second baby. And
tremendously gratifying, I can imagine, to go from a PhD scientist to something where you're saving lives. So you're now a card-carrying tumor immunologist, actually having impact.
People used to use that, by the way, as an insult.
So if you weren't looking at issues of fundamental biology, what else do you think could have kept you engaged over these years? I mean, I know you and I were talking earlier about music, and in fact, I saw this headline, musician scientist wins Lasker Award. So I know you're a musician as well. Do you think music or the poetry you mentioned earlier could have kept your mind as occupied?
No, poetry was just when I was in high school, but no, music, I had a lot of fun playing music. I mean, when I was a postdoc at San Diego, I played with the band, a couple of times a week, and it took its toll, and staying up late and drinking too much beer sometimes, but
did it generate any aha moments?
When you get a group of people and you're really in it, and everybody's clicking, and we have a band now called the Checkpoints, named after checkpoint blockade. Everybody in the band does immunotherapy, but we played together long enough to, we start into something, and Tom Gajewski, who's really the leader of the band, play a lead guitar thing, and he'll look over and nod, and I know that's time when you're communicating non-verbally, just with it's a wonderful sort of thing when you really, have a band really clicking. I really love that. I could have done that. I don't think I would have done well. I'm not that good, but I did have a point when I was a postdoc when the band I played with decided to move back to Texas, to Central Texas, and play the Green Hall, dance circuit, and I said, no, I can't go. Got a day job. I got to stick with that.
Well, I think I'm in particular glad that you did. And it's been a real treat to hear a little bit about your path in science. So thank you very much, and congratulations on the Lasker Award.
Okay, well, thank you very much.
All right.